place where copper, these two transporters are also absolutely required is in the brain. It's sort of playing a role in delivery of copper to the brain. And we'll talk in more detail, so it's a much more complex system, how this transporter works, but both of them are essential for copper delivery to the brain. So in the liver, which is a major homeostatic organ for copper, so when copper gets in, so the first place it's in the liver. So there is another transporter which called ATP7B, which balances copper in the liver and export excess of copper from the cell. And the reason why I like to mention it today, because it's also very important, although a more minor player in brain copper metabolism. So when this transporter doesn't work, so ATP7B, we have another disease which called Wilson disease. And this is also a devastating disorder, even though it does not manifest as early, as remarkably as Menkes disease. So it's still quite devastating. So these patients have also defects in copper transport. So their manifestations are both in the liver, where liver develop inflammation, necrosis, and without treatment it can lead to failure. But there is also a very significant neurological and psychiatric component of this disease, which is very poorly understood. So this patient presents with involuntary movements, tremors, inability to speak, difficulty to swallow. So the challenging, so I work primarily with Wilson disease, and the challenging part for me from sort of when I think about patients, when I think about family, is they actually completely, so the mind is not affected, so they can sort of see what happens to them with every day, and I find this terrifying and very important to deal with. So the reason why I mentioned the Wilson disease, because as I will show you today, even though these two major transporters, 7A and 7 in CTR1 are key, this is a very important modulator, and I will show you how they all three work together to control copper balance. So this is just to get you oriented. So because of the work of many people in the copper field and speakers that you will hear later today, so we know now quite a bit about copper physiology. So we know that copper enters the cell through this transporter I already mentioned, CTR1. It's very important for getting copper into the cell. And there is this various destination for copper to travel. It has to go to mitochondria for energy production. It has to go into the nucleus to support RNA splicing. It has to go to other destinations like lysosomes. But the most important destination for copper inside the cell is this trafficking, we call it trafficking pathway, which is mediated by small protein chaperone. It's a copper carrier, takes copper and delivers copper to two transporters, which I already mentioned, ATP7A and ATP7B. The job of this transporter is to take copper and bring it into the secretory pathway and package copper to this copper-dependent enzyme which participate in myelination of neuron, in neurotransmitter synthesis, in blood vessel formation, collective tissue. And that's why treatment of Menkes disease is so difficult, because even though we deliver copper to the blood and even though we deliver copper to the cell, there still has to be ways to deliver copper to this compartment. So mitochondria is also central, but if copper gets into the mitochondria, as we hear later today, so it provides significant relief to the cell and cell physiology. This is just to illustrate you these two transporters. As I said, we already know quite a bit. CTR1 is a very interesting channel-like transporter, so it has ways to absorb and concentrate copper from the blood in the entryway. So when it concentrates copper here, then it can transfer copper through the channel to the inside of the cell. And that's where protein called ATX1, this little chaperone carrier protein, it takes copper and deliver this to this transporter. So this is just a cartoon. We do not yet know full structure of 7A or 7B, working towards it. So it's a very complex protein, so it takes copper from the chaperone. It then uses energy of ATP to transfer copper through the membrane to these various copper-dependent enzymes, which require for all these reactions I've been mentioning earlier on. So in the brain, I apologize that it's probably very hard for you to see. So in the brain, all these transporters are present in various cells. So the ATP7A, CTR1, ATP7B, they can be found in neurons in various locations. They also can be found in astrocytes. So in the function of these transporters are somewhat different. And more importantly, the distribution of these transporters between different cell types is also different. This is just to illustrate you. So if we look at endothelial cells, so this is cells which are lining blood-brain barrier and important for delivery of copper. So this gray bar is CTR1. You can see there is a lot of it in this type of cells. It's important for entry of copper. So this is 7A. So that is very important for, again, transporting of copper to the brain. Just like I said, 7B is not so critical. But if we look at astrocytes, so this is sort of buffering cells in which control copper levels and supply copper to neurons. So it's a little bit different. So CTR1 is not very high, so but ATP7A and ATP7B are both present. So we still know very little about sort of different cell type and how copper biology works in different cell types. And this is something for us to understand because, of course, every treatment has to take into account how different cells balance their copper homeostasis and how they talk to each other. So this is, I hope you will be able to see. So this is just to show that, so we talk about ATP7A Menkes protein, and we talk about ATP7B Wilson disease protein, and biochemically, these proteins are very, very similar. So if we work with them in a tube, so they do the same reactions, so they use ATP, they transport copper, but in a cell, they're not equivalent, so they cannot substitute for each other. And this is apparent in many, many ways. So one example I already mentioned, that they're present in different cells in different amounts. The other thing which is, so if within the cell, so this is in this white circle, so we're looking at one cell here, this white circle is a nucleus, and you can see that ATP7B in this very characteristic tight location, which is a trans-Golgi network. So it works here to deliver copper to copper-dependent enzymes. Within the same cell, so this in this case we're looking at dopaminergic neurons. So in this very same cells, you can see ATP7B present in very different pattern. So it's in vesicles, it's more distributed. So this different localization tells us that function of these proteins are different. So they work in different places, they do different things. And so this is just cartoons summarizing what they do in this cell type. And this is just one of the examples. And there's so many things remain to be discovered. 7A sits here, and it's deliver copper to this enzyme, which is called dopamine beta hydroxylase. So it packages this an enzyme with copper, so it can then go to secretory granules where it converts dopamine to norepinephrine. So as you know, norepinephrine is very important neurotransmitter. So it in response to signaling, norepinephrine is released. That's how we respond to stress. That's how I released a lot of norepinephrine when my cell phone was stolen. So this is the job of ATP7A. So if ATP7A doesn't work, so there is this very low level of norepinephrine, and there is a catecholamine misbalance. ATP7B in these cells, as I already shows, has absolutely different function. So it sits here connecting, transporting copper in these vesicles to package it, to buffer it, so to make sure that sort of the ATP7A has enough amount of copper but not too much. But it also regulates this reaction, which we do not understand very well, is exporting of soluble form of the DBH into the extracellular milieu. So there is very similar enzyme transporters biochemically, but very different intracellular functions. So we also know way more about the various parameters. So just like I mentioned, so they look very similarly. So it's a protein crossing the membrane. So they have many domains. So they can bind copper. They can hydrolyze ATP. And when we compare Menkes and Wilson disease proteins side by side, so another reason why they're not cannot be replaced by each other, because they Menkes disease work much faster. So it hydrolyzes ATP faster, it releases copper faster. So Wilson disease protein is basically sort of helping other when it comes to measuring, maintaining copper level. So for the rest of the presentation, I like to really focus on how does copper enter the brain. So we know that this is the most critical for these diseases that we were talking today, is to provide copper supply to patients to the brain. So it's become very clear very early on, so that copper enters to the brain in humans and in animals very early. So in this cartoon, so what you're looking, so this is a rat brain, and if you're looking at the panels, so the warmer the color, the more copper you have. And if you look at the day after birth, you can see that when the animal is born, this is true in mice and pigs, it's in human, of course, somewhat different time scale. But very early on, copper levels in the brain is very low. But then there is this very dramatic spike in animals, it's day from 7-14 after birth, when there is massive entry of copper in the brain. So copper gets in the brain, so different regions become populated with copper, and then this process stops. So for example, if you work with animals and we use radioactive copper to measure copper entry, so we know that after 3 weeks after birth, very little copper gets in the brain, and that's also true in humans. So if one does radioactive copper and look at the copper distribution in adults, it's less than 1% sort of after brief dose, which gets into the brain. So this tells us that this particular time period is very important, and we really like to understand better. So what happens during this time? So what allow copper to sort of to populate brain, and why then this processes stop? So how we can augment this pathway to provide better delivery copper to the brain. We also learned that within the brain, there are different areas where copper is really high. So, and two of them I like to highlight here. So this is a cross-section of brain, and you can see this circle, and you can see this really bright spot. So this is so-called locus coeruleus, which is the origin of noradrenergic neurons we were talking about. And you can see the copper is really very high and concentrated here. So which again gives us a clue that not only copper needs to be delivered to the blood, not only copper needs to cross the barriers and get into the brain, it also has to reach the specific destination where it's really needed. What also is interesting, so this is another cross-section of the brain, and you can see this yellow bright color is the certain areas within the brain, they really glowing with copper normally. So copper gets in, and this is supposedly copper storage area where copper could be very, very high. So we'd like to understand how copper is stored and how copper can be released from the storage areas. To do that, we start thinking more and more about copper barriers. So as many of you know, so there is a blood-brain barrier. So this is a complicated structure which surround blood vessels. So this is a cross-section of blood vessels. So this would be endothelial cells, which very tightly connected. So the permeation through these cells very low. There's this pericyte, and there is astrocyte and feed surrounding the structure. In this particular arrangement, very few unwanted compounds or molecule can get through. So in this location, CTR1, we already talked about it, is very important, brings copper from blood into endothelial cells. 7A transported it further, and then this process repeats, so copper can enter the cell. So, and this is just summarized in more simple cartoon here. So CTR1 brings in, 7A gets it further, and then brain parenchyma into interstitial fluid, and then brain parenchyma gets copper. So we always focus on blood-brain barrier first because it is in fact a very important barrier in the brain and critical for transfer of most of the compound. But copper is more complicated. So, and the reason for that, there is a lot of evidence that additional barrier, which is called blood-CSF barrier, may play an important role. So, and one of the reasons for thinking is that blood-CSF barrier plays an important role, and especially during this early time, which we're discussing, is the expression, so the abundance of transporter in this particular barrier. So this is the data from so-called single-cell sequencing of human brain. So, and what you see here in the shape and the size of the dome plexus. 7A is also more abundant, although it's abundantly present in blood vessels as well. And all these other players that we know are important for copper metabolism, they're also very abundantly present in blood-CSF barrier. So that makes us really interested whether or not that's a sort of the gateway we can work with, and whether or not that's where the sort of switching of copper entry during the birth occurs. So this is hypothesis. So, but I just like you to illustrate where it is. So it's located in ventricle. This is just to show how abundantly, this is 7A. So expression, you can see this brown color. So there is a lot of 7A in this barrier. This is one of our other transporters, this copper chaperone. And the interesting part about this barrier that it also very closely located to the storage pool. So this is just to illustrate in more detail how copper gets through the barrier. So it's similar, in a similar way, CTR1 brings copper in, and 7A takes copper out. So the 7B rule was proposed to be transporting copper out of these cells and balance copper. Whether or not that's exact function, we still don't know. So we started working with these barriers just to understand it a little bit better, and I apologize for the lights, so but I hope you can see the main points. So you can see that this is sort of, the cells have very abundant CTR1, which brings copper from the brain side into this structure, choroid plexus. So how copper get in, we still don't understand. And this is something we believe is very important for us to discover, because in all these barriers in copper entry, so the missing player which brings copper into blood-CSF barrier. We also have ATP7A present here. But what's something I wanted to mention in this particular slide, I mentioned earlier that 7A and 7B are not interchangeable, but they actually affect each other. So if 7B, which is a minor player in the brain barrier, is absent, things actually have a significant consequences for copper entry. So in this case, just to show you, so when 7B, very little abundance, much less than 7A and CTR1. But if we delete it, so CTR1 is gone from the plasma membrane, ATP7A is highly upregulated. You see this much brighter red color, and copper is just trapped in choroid plexus. So even though this transporter by itself doesn't play a role in copper entry into the brain, So it's still important of how important transporter functions. So this homeostatic system plays together very well. So what significance of blood CSF barrier also is that it located, so this is triangle that blood CSF area located, is it located in this very copper-rich area, which is a copper storage area. And if we dysregulate function of choroid plexus, in this case, we just dysregulate 7B, even though we have 7A, so we do not have this copper storage, and copper brain get transiently copper depleted. In this case, it's transient because the major player are still there. This has very significant consequences. So even minor copper deficiency, in this case, so what we were looking is just an analysis of different lipids. So we look at the healthy brain early in the development, and we look at the brain with minor copper depletion. So it's much less severe than in Menkes disease. And yet, if you look at this sort of panel where we look at different lipid types, so there is this significant dysregulation of lipid balance. So it is just one of the examples of even minor copper deficiency have significant effect on metabolic processes. So just in final few slides, so I like to show you that copper misbalance has numerous consequences, and copper, slight copper elevation and slight copper deficit lead to cells adapting and changing their property. I already mentioned lipid metabolism. So here I like to show you what happens if we have slight copper elevation in this barrier, in blood CSF barrier. So that's why balancing copper is critical, because here we have only about 50% more copper, so it's not even to full. So, and what we see here in this panel, so this is normal brain, and you can see this sort of very sort of fluffy-looking structure. So this is multiple projections. So blood CSF barrier cells, they're not smooth, so they have numerous projections in order to facilitate transport in and out of the brain. And in the brain, which is, in this case, it's ATP7B, where there is a little bit more copper, so these projections are missing. So there is surface area to exchange nutrients is now significantly less. So, and this happens because we also have a marked decrease in structures in the brain, which called cilia. So cilia have a role in sensing nutrients and also moving cerebrospinal fluid to facilitate the exchange. So usually normal surface of this endothelial cells in choroid plexus, blood CSF area, they're studded with this cilia. They're very abundant because they help cerebrospinal fluid moving. If we have slight copper elevation, so now these structures are poorly formed, they're not very abundant, they're lacking, and this is just to show you electron microscopy picture. This is normal cilia, and this is poor cilia when copper is elevated. So not only we need to provide copper to the brain, provide it through the right route, but also it has to be exactly right. So which is, of course, as you know, a main challenge. So this is just to finish up to show you what we learned so far. We learned that if we have copper elevation, which is relatively mild, so this is various proteins which are affected, and these proteins are participate in cytoskeletal structures. So they support cell shape and morphology. So, and if copper is elevated, you can see that normally looking microtubules, so they form structure for cilia and protrusions, so they look defective, so they're abnormal. So, and I will finish with the major player for this copper delivery to this microtubule. We think that this small copper carrier requires for bringing copper towards these tubules and bring them up. So we were able to show that actually localized together in the cilia. And we believe now that there is a lot to learn about how all these transporters work together to balance copper, to provide it to the places where it is needed, and to maintain it in the exactly right proportion. So this is just to summarize. So we think choroid plexus, in addition to blood-brain barrier, very important. So it facilitates copper delivery to the brain. We do not yet know whether or not it plays a critical role during early period of time, but its location close to these copper storage pools suggests that it may be very important. And ATP7A and CTR1 are essential players here, but I would like to mention that 7B, which balances copper, which does not allow copper elevation, is also very critical. So this is the team I would like to acknowledge, and funding from NIH. And thank you for your attention, and it's really the pleasure and honor, and I hope this work will continue.
If anybody has any questions, that's some time that you ask
Svetlana, it was a wonderful talk. I learned a lot. I always do whenever you present. Thank you. So Svetlana, my question is, so you described a few areas in the brain where copper is stored. So do you know the areas and the type of cells where copper is stored?
So the major location of copper stores are in ventricle area, so in surrounding area. They're thought to be specialized astrocytes.
Yeah, because the translator is not, yeah.
So these are ventricle areas, so the cells which are lining this area are specialized astrocytes. It is not yet clear what sort of the storage moiety is. From my work, we know so it may be metallothianins may be involved. So, but how they release copper is not clear. But the importance of the location, it's not only they're close to entryway, but they're also very close to neuronal stem cells. So, and the hypothesis is that this critical location of copper stores near the stem cells is sort of necessary in case of brain damage, in case of some deficiency. So, and that's how copper is being recruited from the stores.
Thank you.